In this experiment, two fruit flies, a female virgin with white eyes and a male virgin with wild-type red eyes, were used to determine genotypes and phenotypes of their progeny. This experiment was conducted over a 5-week period and produced an F1 and F2 progeny. The genotypes and phenotypes of the progeny were recorded and compared. To begin, the parental flies were placed into vials with a media to ensure that the flies were able to survive and reproduce efficiently. After a progeny was produced and were in their larvae state, the parental flies were removed from the vial using fly nap, which is a safe way to anesthetize the flies for evaluation, and the larvae were left to hatch. After hatching, the progeny flies were observed under a microscope to determine the sex and eye color. A male and female from the F1 progeny were then mated to produce an F2 progeny. After larvae appeared in the vial, the parental flies were removed using the fly nap, and the larvae were left to hatch. After the F2 progeny hatched, fly nap was administered to determine the sex and eye color of each fly. Drosophila Melanogaster, also known as a fruit fly, is a commonly used organism for lab experiments. Scientists use fruit flies in many experiments because they are known as a model organism. Fruit flies are model organisms due to their fast gestation periods and are easily acceptable to live in a laboratory setting. The fruit flies used in this lab survived off of a media. The media is made using water, fly medium, and yeast grains. It is important to know how to find the sex of a fruit fly. The sex of a male fruit fly can be determined by observing the front legs and noticing the sex combs. Males use their sex combs to grasp the female's abdomen and genitalia and to spread their wings prior to mating. Female fruit flies do not have sex combs on their legs. Male fruit flies also have a darker pigment on their abdomen, while females do not. A wild-type fruit fly consists of a tan body with red eyes. In this experiment, the mutant fruit fly that was used consisted of a tan body with white eyes. The trait for red eyes is a dominant trait. A dominant trait is a trait that will show regardless of whether or not the organism carries one or two alleles for that trait. The trait for white eyes is an X-linked trait. An X-linked trait is a trait that is carried by the determining chromosome X. The first mating was conducted between a white-eyed female fly and a wild-type red-eyed male. It was hypothesized that all of the females would be wild-type and all of the males would be wide-eyed, due to the dominant trait being the wild-type and the white-eyed trait being X-linked. The F1 progeny was then mated to produce an F2 progeny. In the second mating, a white-eyed male was crossed with a wild-type red-eyed female. It was hypothesized that there would be a 1-to-1 ratio of all phenotypes within the progeny due to the dominant trait being carried by a heterozygous parent. To begin this experiment, fly nap anesthetic was administered to the parental flies. Once asleep, the parental flies were distributed, and the sex and phenotype of each were determined by an observation under a microscope. A media was created and placed within a vial where the flies would reproduce. The parental flies were left in the vial for 1 week in order to reproduce. After 1 week, fly nap was administered to the parental flies so that they could be removed efficiently from the vial and only the larvae would remain. The parental flies were placed into an ethanol bath where they were then euthanized. The larvae were left to hatch for 1 more week. 7 days later, fly nap was then administered to the F1 progeny in order for them to be easily handled and observed for their sex and phenotype. After the progeny was observed and the results were recorded, 5 females and 5 males from the F1 progeny were placed into a vial to create an F2 progeny. After 1 week, FlyNap was administered to the F1 progeny parental flies so that they could be removed and only the larvae would remain. 1 week passed, and the FlyNap was then administered to the F2 progeny that was created in order for them to be easily handled and observed for their sex and phenotype. After the F2 progeny was observed and the results were recorded, the F2 progeny was properly disposed of in an ethanol bath. This table demonstrates the F1 progeny that was created from the first mating between a white-eyed female and a wild-type male. The data concludes that there were a total of 73 flies produced, 36 of which were males and 37 that were females. All 36 of the males were white-eyed, and all 37 of the females were wild-type red-eyed flies. This table demonstrates the F2 progeny created from the cross between a white-eyed male and a wild-type female. The data concludes that there were a total of 32 progeny, 12 of which were males and 20 that were females. There were 7 wild-type red-eyed males and 14 wild-type red-eyed females. The data also concluded that there were 5 white-eyed males and 6 white-eyed females. This experiment was conducted in order to observe how a mutation is passed on from a parent to an offspring. Fruit flies were used to observe how a mutant trait and a dominant trait affected a progeny. The fruit fly was used due to its rapid reproduction cycle and the ability to live in a laboratory setting. The mutation being studied in this experiment was a white-eyed mutation. A non-mutated, also known as a wild-type, fruit fly has red eyes. The first mating occurred between a wild-type male and a white-eyed female fruit fly. The progeny of the first cross were counted, sexed, and evaluated for mutations. The data concluded that all of the males produced were white-eyed and all of the females were wild-type. This occurred due to the male offspring only receiving one X chromosome and the females receiving 2. The hypothesis was correct to the white-eyed mutation being sex-linked and the wild-type eyes to be dominant. 5 females and 5 males from the F1 progeny were left in a vial to mate for 1 week to produce an F2 progeny. The progeny were counted, sexed, and evaluated for mutations. The data concluded that there was a mixture of males and females with both red and white eyes. The hypothesis was that there would be an equal number of progeny for each gender associated with each mutation. Although there was a mixture of both males and females having both red and white eyes, the hypothesis was not supported by the results. There was not an equal number of progeny for each gender associated with each mutation. The results could have been affected due to an exponential error. The error that could have occurred may have been that many of the F2 progeny flies died in the media and were not able to be counted, sexed, or evaluated for mutations. This could have altered the number and ratios of each type of fly. This experiment concluded that the white-eyed mutation is sex-linked compared to the dominant trait of red eyes.